This is the video for Lab 11, Meiosis and Genetics. In the last lab, we went over the process of mitosis. Mitosis is a type of asexual reproduction where one parent cell divides into two identical daughter cells. Meiosis, on the other hand, is a specialized type of cell division which will produce 4 daughter cells, and those cells will be haploid. And haploid means they will have half the number of chromosomes as the original parent cell. And this is a special type of cell division that is involved in sexual reproduction. So in sexual reproduction, you start here with the diploid adults. So all the cells in the adult are diploid. That means they have 2 copies of every chromosome. Then the process of meiosis is going to create haploid cells, and we call those the gametes, and those are the eggs and the sperm. So that when a sperm fertilizes the egg, then you go back to having a diploid cell, and then all the body cells are derived from that fertilized egg. So that's the sexual reproduction cycle, starting with a diploid organism, diploid parent cell, go through the process of meiosis, generate haploid gametes, and then the gametes fuse together to form a diploid cell, which generates the organism. When going through meiosis, there's some important vocabulary that you need to know, and this vocabulary relates to the different chromosomes. The first term is homologous chromosomes, and homologous chromosomes are chromosomes in your cells that have the same size, same centromere location, and contain the same types of genes. And you got one set of your chromosomes from your father, one set from your mother. So, for instance, this could be chromosome you got from your father, and this is condensed chromosome. So that is representing those chromosomes. But this is one chromosome you got from your father, and you also got one from your mother. So these chromosomes have the same size, same centromere location, and they contain the same types of genes. During S phase, the chromosomes are going to be duplicated. So again, this is one chromosome, but when it duplicates, then the duplicated chromosome will be held together by the centromere. So sister chromatids are identical copies of your original chromosome. And then also with the maternal chromosome, it will be copied, and then you will end up with sister chromatids for your maternal chromosome. And this is considered one chromosome because they're attached by a centromere, even though it is 2 separate chromatids. And together, your maternal chromosome with sister chromatids and your paternal sister chromatids, together they are homologous chromosomes, and all 4 of them are called a tetrad. Meiosis involves 2 rounds of cell division. And the first round of cell division is called meiosis 1, and that's going through prophase, metaphase, anaphase, and telophase. So we refer to those phases as prophase 1, metaphase 1, anaphase 1, and telophase 1. At the end of telophase, the cell goes through cytokinesis, and you end up with 2 daughter cells. Then in meiosis 2, both of those daughter cells go through a second round of cell division. So we call these prophase 2, metaphase 2, anaphase 2, and telophase 2. And at the end of telophase 2, each of those daughter cells divides. So you will end up with 4 gametes at the end of meiosis 1 and 2. During meiosis, what you want to focus on is what is happening to the chromosomes. So during meiosis 1, and again, meiosis 1 is composed of prophase 1, metaphase 1, anaphase 1, telophase 1. During prophase 1, as these chromosomes are condensing, the tetrads are held together, and that is where you can get crossing over. So crossing over can occur during prophase 1. Then during metaphase 1, you need to know what is lining up along the metaphase plate, and those are the tetrads. Remember, a tetrad, that involves those two homologous chromosomes that are composed of two sister chromatids each. Then during anaphase, what is separated? Those homologous chromosomes are separated from each other. So the tetrad is basically split in half, and the homologous chromosomes are pulled to opposite ends of the cell. Then you go into telophase 1, where the cell divides, and you will end up with two daughter cells that will head into prophase 2. The next stage is meiosis 2, and meiosis 2 involves two cells going through the process, because remember, at the end of meiosis 1, you generated two daughter cells. In metaphase 2, you need to know what is lining up along the metaphase plate, and those are the chromosomes. And each chromosome is composed of two sister chromatids that are held together by a centromere. Then in anaphase 2, the centromere is going to split, and the two sets of sister chromatids are pulled apart. So this is the stage where the sister chromatids are pulled apart. And when you get to telophase 2, both of these cells are going to divide, so you will end up with 4 gametes. And those gametes will be haploid. They will have half the number of chromosomes as the original parent cell. The karyotype is a display of all of an individual's chromosomes, and this is used to identify genetic disorders that are caused by differences in chromosome number. In order to see the chromosomes, we need to be able to look at them when they are in the condensed form. So usually these chromosomes are taken from a metaphase cell. So a metaphase cell, this is when the chromosomes are nice and condensed, but remember, each chromosome is composed of two sister chromatids held together by a centromere. So an actual karyotype would look like this. This is an actual karyotype. So the chromosomes are condensed, and you have two sister chromatids held together by centromeres, so it's relatively easy to see them. In the human cell, we have 23 sets of chromosomes. So we have the first 22 are just named by their size, or numbered by their size. Chromosome 1, 2, 3, 4, 5, and so on, all the way till you get to 22. And the chromosomes get progressively smaller. Each type of chromosome, so for instance, chromosome number 1, you got one chromosome for your father and one chromosome for your mother. So that's your set of homologous chromosomes. The last set of chromosomes are called the sex chromosomes, and those are the X's and the Y's. This individual has 2 X chromosomes, so it is a female. If an individual has 1 X and 1 Y, it would be a male. So any individual that has a Y chromosome, no matter how many X's, will be male. And the Y chromosome is much smaller than the X chromosomes. Genetic disorders that are caused by an abnormal number of chromosomes occur because of nondisjunction. Nondisjunction is when the chromosomes fail to separate during anaphase. So remember, during metaphase, you have chromosomes lining up along a central axis, and here you have 2 sister chromatids held together by centromeres. Normally what would happen is the centromere would split, so you get one set of sister chromatids going to one end of the cell and the other set going to the other end of the cell. In nondisjunction, the centromere would fail to separate, and then both of these sister chromatids would go to one end of the cell. And this would result in gametes that have abnormal number of chromosomes. In meiosis, because the process involves 2 rounds of cell division, there are 2 points where non-disjunction can occur. So here's a diagram. You have meiosis 1, meiosis 2, and in this case, non-disjunction is happening during meiosis 1. So during anaphase 1, this tetrad fails to separate, and both of those homologous chromosomes go to this daughter cell. So as you go through the process, you end up with 4 gametes, and they all have abnormal number of chromosomes. So the haploid number should be 2, but half of the gametes will have 3, and the other half will have 1. You can also have non-disjunction during meiosis 2. So meiosis 1 occurs normally, but during meiosis 2, the sister chromatids fail to separate, and this will generate 1 gamete with 3 chromosomes, 1 gamete with 1 chromosome, but then these 2 gametes will have the normal haploid number. The other part of the lab is going to deal with genetics, and the father of genetics is Gregor Mendel. He was a monk who lived in the 1800s, and he investigated how information for different traits is transmitted through generations. When doing problems with genetics, it's really important to understand certain vocabulary terms. So the first one we'll go through is genetics, and as I said before, this is the study of how information is passed from one generation to another. So of course, now we know that the form of this information is DNA. But back in the 1800s, Gregor Mendel, he had no idea. They didn't know what DNA was. So he used the term allele as an alternate form of a trait. And so of course, we know that this is a gene now. But the term he used was allele. So we still use this, but just keep in mind that an allele is basically a gene. So it's an alternate form of a trait. Phenotype. Phenotype is the physical expression of a trait. So it's what you actually see. Is the plant tall? Does it have purple flowers? Does the person have brown eyes? It is the physical expression of the trait. So phenotype, PH, physical. The genotype is your genetic makeup, so genogenetic. So these are the actual genes that you have. And because you got genetic information from your father and your mother, you will have 2 alleles for every trait. You get 1 set of alleles from your mother, 1 set from your father. With those alleles, if they are homozygous, that means they are identical. So that means a plant could have 2 tall alleles or 2 short alleles, or a person can have 2 brown-eyed alleles or 2 blue-eyed alleles. Heterozygous, hetero means different. This means the individual has 2 different alleles for a trait. So that would be 1 tall allele and 1 short allele, for example. Now the behavior of the alleles, this is represented by dominant and recessive. An allele is dominant if you need only 1 of those alleles present to get the trait. Recessive, a recessive allele means that you only get the trait if you have 2 alleles. So the only way you get a recessive trait is if an individual is homozygous and has 2 identical alleles for that trait. Punnett squares are very useful tools in genetics because Punnett squares can predict the results of a cross. So when you have 2 parents, you can use a Punnett square to figure out the probability of the genotypes of the offspring. And if you know the genotypes, you'll be able to figure out the phenotypes. So again, Punnett squares give you the probability of the genotypes of the offspring. To do a Punnett square, there are 4 basic steps you need to follow. The first step is to determine the genotypes of the two parents. So remember, genotypes, those are the two alleles that each parent has. The second thing you need to do is to determine what types of gametes those two parents will produce. So this would be the result of meiosis. So as those parent cells go through the process of meiosis, what types of gametes in terms of what allele each gamete will have. Once you have determined the types of gametes, then you just need to fill out the matrix of the Punnett square. Once the Punnett square is filled out, then you can determine the potential genotypes that the offspring can have, and from the genotypes, you can figure out the phenotypes. This is an example of what a Punnett square looks like, and I'll go through it in more detail. But here are your two parents. So this is the female, this is the male. You figure out the genotype, that's the first thing you do. Then you figure out what gametes each one will produce, and this is a result of meiosis. Once you have figured out the gametes, then you fill out the Punnett square. And the Punnett square, this represents 100% of the potential genotypes of the offspring. So each box, in this case, represents 25%. And once you figure out the genotype, then you can figure out the phenotypes of the offspring. One type of Punnett square involves a monohybrid cross, and mono means one. So this is a Punnett square that analyzes one trait in a cross. And again, this is a Punnett square, so it will determine the genotypic and phenotypic probability for just one trait. So an example of this could be looking at the one trait of flower color in the pea plants. In the pea plants, there are two alleles. One is a purple allele, one is a white allele. And the letter they have chosen is P for purple because purple is dominant, so that is a capital letter. Lowercase p is for the white allele. So for a plant to express purple flowers, it needs only one dominant allele. For a plant to express white flowers, it needs to have two recessive alleles. It needs to be homozygous for the recessive allele. The two parents, so the first stage is figuring out the genotypes of the two parents. So we've given you two parents. One is homozygous dominant, the other is heterozygous. So first process in the Punnett square, figure out the genotypes of the parents. Second process of the Punnett square is figuring out the two types of gametes that each parent can produce. This parent, half of the gametes are going to have that green dominant allele, and half of them will have that purple allele. The other parent, half of the gametes will have the dominant allele, half will have the recessive allele. So once you've figured that out, you can set up the Punnett square. So these are the alleles for that first parent, and they're just colored differently so that you can see them. And then this parent, the alleles go here, dominant allele here, and recessive allele there. So then you just fill out the Punnett square. So these alleles go across this way. Put these alleles down that way. And now you can analyze the Punnett square. Once you have figured out the genotypes of the parents, figured out and filled out the alleles for the gametes for both of those parents, now it's time to analyze the results. And what you can see here is that you have only two options for genotypes for the offspring, homozygous dominant and heterozygous. And with the ratio, you have 50% will be homozygous dominant and 50% will be heterozygous. So if you want to use this as a ratio, it would be 1-to-1, or you can refer to it as 50%. 50% homozygous, 50% heterozygous. So that's the genotype. For the phenotype, you can figure that out by looking at the genotype. So a homozygous dominant will be purple, and a heterozygous will be purple. So 100% of these offspring have at least one dominant allele. So 100% are going to be purple. And when you analyze this, 100%, the ratio is 1 to 0. Nothing will be white. 100% will be purple. Another type of cross you'll have to be able to do is a dihybrid cross. And a di means two, so this involves two traits that are going to be evaluated. And the important thing about this is that those two sets of alleles for those two different traits, they're going to sort independently during meiosis. This is an example of a dihybrid cross in pea plants. In pea plants, you are looking at two different traits. One trait is the color of the peas, the other trait is the texture of the peas. And for this pea plant, you have two parents that are heterozygous for the traits. So heterozygous for texture, round is dominant, so use the capital R, wrinkled is recessive, so you use the lowercase r. Color, yellow is dominant, so it has the capital Y, and green is recessive, so it is the lowercase y. So in this case, both parents are heterozygous. So you have that's one parent, and this can be the other parent. Okay, so once you've decided the genotype for each parent, now the hard part in a dihybrid cross is figuring out the gametes that each parent produces. And because these parents have the same genotype, they will produce the same types of gametes. So figuring out the gametes. Remember that alleles sort independently. So during meiosis, when you're going from diploid, having two copies of every, two alleles for every trait, you're going to go haploid, where you're only going to have one allele for each trait in the gametes. So as you can see, every gamete has one R and one Y. But determining which R goes with which Y, you can use FOIL. First, outside, inside, last, or however you want to do it. So during meiosis, this dominant R is going to go half the time with the dominant Y. And then half the time during gamete formation, it's going to go with a lowercase y. So that dominant R will go half the time with the uppercase y, half the time with the lowercase y. The little r is going to go half the time with the uppercase y, half the time with the lowercase y. So you're going to get that little r, recessive r, with the dominant y, and the recessive r with the recessive y. And these will be your 4 gamete options for that one parent and also for the other parent because they have identical genotypes. Now in this case with a dihybrid cross, because you have an option of 4 gamete types for each parent, that means your total square is going to have 16 options inside. So analyzing the results can get a little tedious, so just be very careful that you are counting all the different potential genotypes of the offspring correctly. For your lab report, the first experiment is going over the PCR and electrophoresis that you would have done the previous week. So what you would have done in lab is you would have extracted some of the DNA from your cheek cells and then run it through a PCR reaction to generate lots of copies of the DNA. Once you had done that, then you would have taken that DNA and loaded it into a gel, which would have separated out fragments of DNA by size. This is an example of a gel that you would have seen. And for this gel, you would have loaded the DNA here into those depressions. Those are called wells. and then the electricity would have moved the DNA in that direction. So these bluish bands are bands of DNA of different lengths. And remember, the shorter the DNA is, the further it would move through the gel. For your gel, you were looking at the ALU gene, and to determine if you are homozygous positive for the gene, homozygous recessive for the gene, or heterozygous. So these are the banding patterns for homozygous positive, homozygous negative, and heterozygous. These are 3 student samples. So this is student 1, student 2, student 3, student 4. So what I want you to do is to look at the banding pattern and determine the genotype for each student. Experiment 2 on your lab report involves meiosis, drawing in the missing chromosomes in the mitotic process. So here it is a review of what is lining up and what is separating during the 2 different stages of meiosis. The third exercise is on karyotyping. In karyotyping, I want you to go to this website, which will look like this, and you go through an activity where you finish the karyotype for 3 different patients and determine what genetic disorder they have. So experiment 4, the taste papers, you are not going to do that, but experiment 5, the human traits, you are going to do this. So for this, you are going to determine your own phenotype and, if possible, determine your genotype. So as an example, tongue rolling. So tongue rolling, this person cannot do it. This is a picture of what tongue rolling looks like. If I were going to do this, I would determine if I'm able to roll my tongue or not, and I am able to roll my tongue, so this is my phenotype, and the trait is a dominant. So since I am able to roll my tongue, I know I have at least 1 dominant allele, and fortunately, I know that my father cannot roll his tongue, so he must be a homozygous recessive, which means that I must be heterozygous. Because my father is a homozygous recessive, so I had to get a recessive allele from him, and since I have the phenotype, that means I am heterozygous. All right, other traits. Tongue folding, this is what tongue folding looks like. Dimples, so you know what dimples look like. And widow's peak, so that's what a widow's peak looks like. Some other traits you will also look at. Hitchhiker's thumb. Hitchhiker's thumb is where your thumb bends back significantly. Thumb crossing. This is when you just clasp your hands together. Which thumb is on top? So having your left thumb on top is a dominant, and having your right thumb on top is recessive. Then attached earlobes versus detached earlobes. Attached earlobes, the earlobe goes straight into the neck, where detached, you have a separation of the earlobe from the neck. And then the last one, freckles. Freckles are dominant.